Welcome to the peaceful space of sleep-friendly science, where tonight we will listen for the whispers of ghosts, not in an old house, but in the ancient living architecture of our own DNA. Here in the quiet of this moment, let the outside world fade away. There is nothing you need to do, nowhere you need to be. Simply allow your body to grow heavy and soft, and your mind to become curious, as we explore the story of how ancient pandemics forever shaped the human story. Now settle in, take a deep breath, and let the soft sounds guide you as we begin tonight's journey into the unknown. Section 1: The Living Library of You. Let us begin our journey tonight by floating down into the quiet and intricate universe that is you. Picture yourself not as a solid form, but as a galaxy of trillions of stars, and each star is a single living cell, humming with a silent, purposeful light. Within every one of these cells is a sacred central chamber, the nucleus, and it is there, coiled in threads of impossible beauty, that we find the book of life, your genome. For a long time we thought of this book as a simple blueprint, a straightforward, practical manual for building a human. It contained the recipes for the proteins that make your eyes a certain color, the instructions for the collagen that gives your skin its softness, the code for the hemoglobin that carries the breath of life through your veins. We saw it as a text written in a simple four-letter alphabet TC, and we believed that the author of this text was simply you, your inheritance, your personal story. But tonight, I invite you to see this book in a new light. Let us set aside the idea of a simple blueprint and imagine it instead as a living library, or a vast ancient chronicle. The story it tells is not just of you in this moment. It is an epic passed down through a chain of hands that stretches back into the mists of time, beyond the first glimmer of humanity, to the very dawn of life itself. Every chapter, every verse, every letter has been copied, edited, and passed on from your parents and their parents and all the ancestors who came before them. This chronicle is the story of their survival. It tells of the famines they endured, the climates they adapted to, and the evolutionary pressures that shaped them. It is a story written in the ink of experience, and you are its current living custodian. But when we look closely at the pages of this ancient book, we find something strange. It is not a clean, pristine copy. The ink is smudged in places. There are notes scribbled in the margins in a language we don't immediately recognize. Entire passages seem to have been crossed out, while others are pasted in, as if from another book entirely. The library is haunted. It is filled with the echoes of other voices, the shadows of uninvited authors who have left their mark upon your text. Who are these ghosts? Who are these other authors who have written themselves into the story of you? The answer is as profound as it is intimate. The ghosts are viruses. For as long as your ancestors have walked the earth, they have been in a silent, locked embrace with these microscopic entities. It is the oldest and most intimate of relationships, an eternal dance between predator and prey, between the host and the uninvited guest. A virus is a strange and beautiful thing. It is pure information, a small packet of genetic code whose only purpose is to find a home, a library where it can be copied. Cannot live on its own. It needs a host, needs your cells. Imagine an ancient pandemic, a wave of sickness sweeping through a population of our distant ancestors. This was a war fought not with weapons of stone, but with the subtle tools of molecular biology. It was a battle for information, a struggle for control of the cell's copying machinery. Most who were infected either perished or fought off the virus and survived, their bodies carrying only the memory of the infection in their immune systems. But something else could happen. Something rare and strange and transformative. Sometimes the virus wasn't just in the cells of the body, the lungs, or the blood. Sometimes it found its way into the most sacred cells of all, the germline. These are the cells that are destined to become the next generation. The sperm and the egg. When a virus writes its story into the book of a germline cell, it does not die when its host dies. It does not vanish when the infection is cleared. It has found a path to immortality. It has stitched its own genetic code into the chronicle of humanity, becoming a permanent passenger, a new sentence in the book of life that will be copied and passed down through all subsequent generations. The ghost has entered the machine, and once it is there, it is no longer just a memory of a past infection. It is now a part of us, an indelible, inherited scar from a battle fought by an ancestor you will never know, an ancient pandemic whose echoes still whisper within you tonight. This is where our story begins. Section 2. An invasion of the germline. To understand how a fleeting infection can become a permanent part of our human story, we must first understand the nature of the invaders themselves. Not all viruses are created equal. Many are simple assassins getting in, making copies, and getting out. But our story tonight centers on a special, and in many ways more sophisticated class of virus. They are the master forgers, the smugglers of information. They are the retroviruses. A normal virus is like a pirate who captures a factory and forces the workers to produce more pirate ships. It brings its instructions and the cell follows them. But a retrovirus is far more subtle. It is not content to simply give orders. It seeks to rewrite the factory's own instruction manual. It wants to add its own chapter to the book of life. Most life, including you, operates on a simple flow of information. Your DNA, the master blueprint, is transcribed into a temporary message, a molecule called RNA. This RNA message is then read to build a protein. It is a one-way street from DNA to RNA to protein. This is the central dogma of life. Retroviruses, however, are renegades. They defy this sacred rule. Their genetic code is made of RNA, not DNA. And they carry with them a secret tool, a magical enzyme called reverse transcriptase. Think of it as a molecular scribe with a near supernatural ability. It can listen to the fleeting temporary whisper of an RNA message and translate it backward, forging it into the solid, permanent, double-stranded language of DNA. The retrovirus can turn its own ephemeral message into a piece of text that looks just like our own. It translates a whisper into a stone carving. Now let us return to our ancient ancestor, living in a world filled with these subtle invaders. We must make a critical distinction, one that is key to our entire story. We must distinguish between the two great kingdoms that exist within every living creature: the somatic kingdom and the germline kingdom. The somatic kingdom is the body, you know. It is the vast, bustling, mortal empire of your flesh and blood. Your lung cells, your skin cells, your heart cells, your brain cells, these are all somatic. They are loyal, hardworking citizens, but they are mortal. Their story ends when you do. An infection in this kingdom, no matter how severe, is a personal struggle. If a retrovirus writes its code into the DNA of one of your liver cells, that altered cell may live and divide, but its story is contained within your liver. It cannot be passed on. The invasion dies with the king. But then there is the other kingdom, the germline. This is not a vast empire, but a tiny, sacred, and immortal river that flows through the heart of the mortal kingdom. The germline cells are the sperm and the eggs. They are the keepers of tomorrow. They do not build the body of today. Their sole purpose is to carry the chronicle of life, the book of your DNA, into the next generation. They are the precious cargo, the living message passed from ancestor to descendant across the abyss of time. An invasion of the somatic kingdom is a battle. An invasion of the germline is a coup. Now imagine the perfect heist, a sequence of events so improbable it borders on the miraculous. Our retrovirus infects an ancient human ancestor. It moves through their body a silent, invisible tide. By sheer chance, it evades the roaming patrols of the immune system. And by an even greater stroke of luck, it finds its way into the protected sanctum where the germline cells are forged. Here it infects one of these precious cells, a cell destined to one day create a new life. And now the retrovirus deploys its magic. The reverse transcriptase scribe gets to work, listening to the viral RNA and forging it into a perfect strand of DNA. This new viral DNA is now indistinguishable from the host's own genetic material. It is a wolf in sheep's clothing. Then comes the final audacious step. This newly forged viral DNA must find a way to insert itself into the host's own chronicle. Guided by another of its enzymes, it finds a place in the long coiled thread of our ancestors' chromosomes and, with a molecular snip and stitch, it weaves itself into the text. It is no longer a foreign object. It is a new paragraph, a new verse, secretly stitched into the sacred book. The heist is complete. The virus is now a silent passenger. It may not even be active. It has simply become part of the background text. And then the final stroke of fate. That one specific altered germline cell, that one egg or one sperm carrying the secret viral message, must be the one that partakes in fertilization. It must be the one that grows into an embryo, a child born with the ghost of the virus in every single cell of its body, including its own germline. This child then grows up and has children of its own, and the viral sequence is passed on again, and again, and again, through countless generations, through ice ages and migrations, through the rise and fall of civilizations. The echo of that one ancient infection is faithfully copied, a permanent tattoo on the soul of a species. The virus has achieved what all life strives for: immortality. It has become what we now call an endogenous retrovirus, an invader that now resides within, a ghost in our machine. Section 3: Fossils in the Darklands. The audacious heist is complete. A viral whisper from a forgotten eon has been carved into the stone of our DNA. But where do we find these ghosts today? If our genome is a vast chronicle, on which page is this secret script written? For years, as we first learned to read our own genetic book, we saw no sign of them. We meticulously read the familiar chapters, the 20,000 protein-coding genes that build and run our bodies. And in those well-lit pages, the story seemed coherent, purely our own. The secret was hidden in the parts of the book we couldn't read. It was in the vast, mysterious 98% of our genome that we, in our ignorance, dismissed as junk. We saw it as evolutionary clutter, the accumulated nonsense of a billion years of copying. We called it the genetic darklands. We now know that this term was a confession of our own lack of understanding, not a description of the landscape itself. For these darklands are not empty. They are an archive, a graveyard, a vast sprawling museum of our deepest history. And it is here, in the supposed junk, that we find the viral fossils. When we talk of these ancient viral invaders, we are not talking about complete, living viruses lurking in our DNA. The vast majority of them are long dead. They are true fossils, as real and as ancient as the mineralized bones of a dinosaur. Over the millions of years they have traveled with us. They have been weathered by the relentless forces of time. Random mutations have chipped away at their code. Parts have been lost, deleted. Entire genes have been broken, silenced, rendered unreadable. They are fragmented, shattered echoes of the vibrant, dangerous entities they once were. Scientists who hunt for them are like genetic paleontologists. They are not looking for a living creature, but for the unmistakable patterns of a creature that once lived. They sift through the long, seemingly random sequences of the Darklands, searching for telltale signatures. It is like finding a single, strangely shaped vertebra in the dust, and knowing you have found the trace of a great beast. What are these signatures? A retrovirus, even a broken one, has a characteristic anatomy. It has a kind of genetic skeleton. A typical retrovirus carries a set of core genes. There is a gene for its structural parts, its viral body. There is a gene for the magical enzymes it uses, like the reverse transcriptase scribe. And there is a gene for its outer envelope, the key it uses to unlock our cells. In these ancient fossils, we can often still see the ghostly outlines of these genes, degraded and misspelled by time, but still recognizable. But the most powerful signature of all is found at the beginning and the end of the viral code. These are known as long terminal repeats or LTRs. Think of them as the unique, ornate bookends that the virus places around its text before inserting it into our own. They are long, highly repetitive sequences that are a dead giveaway, even if the viral genes between them are completely eroded and unreadable. The presence of these two matching LTR bookends in the middle of our own DNA is like finding a pair of beautifully carved, foreign handles on a drawer in your own kitchen. It is definitive proof that something from the outside was inserted here. And so, armed with this knowledge, the genetic paleontologists began to dig. And what they found was not a few isolated fossils. What they found changed our understanding of who we are. They found a graveyard of almost unimaginable scale. The Darklands were teeming with viral ghosts. It is difficult to overstate the sheer scale of this discovery. It is not one or two ancient viruses that have become part of us. It is thousands, tens of thousands. It is estimated today that a staggering 8% of the entire human genome is not human in its origin at all. It is the fossilized remains of these ancient retroviral infections. Let that sink in for a moment. Feel the quiet weight of it. 8% of the very code that makes you is a graveyard of ancient viruses. That is more than 4 times the amount of DNA that makes up all of our 20,000 protein coding genes combined. The ghosts are not a minor character in our story. In terms of sheer volume, they are one of the main authors. The Chronicle of You is written on pages that are overwhelmingly composed of the remains of our oldest and most persistent enemies. Each one of these fossils, what scientists now call endogenous retroviruses or ERVs, is a monument to a pandemic that our ancestors survived. It is an evolutionary scar, a molecular trophy from a war fought in a time before memory. The Darklands are not junk. They are the battleground. And by learning to read these fossils, we can begin to piece together the history of the epic, silent, and unending war that has shaped us. The evolutionary arms race between our lineage and the viral world. Section 4. The Red Queen's Race. Finding these viral fossils in our DNA is like an archaeologist finding the buried ruins of a fortress. It tells us that a great battle was fought here. But it also raises a deeper question. How did this battle, this ancient and unending war, shape the fortress itself? How did the constant pressure from these viral invaders sculpt the very genes of our ancestors? The story of the fossils is only one half of the story. The other half is written in our own living genes. It is the story of a desperate, high-stakes evolutionary arms race. This is often called the Red Queen's Race, named after a character in Lewis Carroll's Through the Looking Glass. The Red Queen tells Alice, "Here you see it takes all the running you can do to keep in the same place." This perfectly describes the relationship between a host and a virus. For millions of years, our ancestors and their viral foes have been locked in this race. The virus evolves a new way to invade our cells, and our genome must evolve a new way to defend itself. We run as fast as we can, not necessarily to win, but simply to survive, to stay in the game. And the epicenter of this race, the part of our genome that has been most relentlessly forged and reforged in the fires of ancient pandemics, is our immune system. Your immune system is a library of defenses. It has genes for creating armies of specialized cells that hunt down and destroy invaders. It has genes for building molecular alarms that sound the warning when a cell is breached. And it has genes that create gatekeepers, proteins that sit on the surface of our cells and try to block viruses from entering in the first place. When we look at these immune genes across the whole of the human population, and when we compare them to the genes of our primate cousins, we find something remarkable. They are some of the most rapidly evolving, most diverse, and most dynamic genes in our entire genome. They are constantly changing, constantly being updated. Why? Because the viruses are constantly changing too. Let's imagine one of these gatekeeper proteins. Let's call it TRIM5 alpha. This is a real protein in our cells, and its job is to be in bouncer. It recognizes the inner structure of a retrovirus shortly after it enters the cell and flags it for immediate destruction, stopping the invasion before it can even begin. Now imagine an ancient pandemic. A new retrovirus emerges that has a slightly different inner structure, one that our ancestors' TRIM5-alpha can't recognize. This virus is now invisible to the bouncer. It can slip past the defenses. Individuals with the old version of the TRIM5-alpha gene are highly vulnerable. Many of them perish. But by sheer chance, in the endless shuffling of genetic information that happens with each generation, a few individuals are born with a slightly mutated, slightly different version of the TRIM5-alpha gene. Their bouncer protein has a new shape. And this new shape, by pure luck, happens to be able to see the new virus. These individuals can fight off the infection. They survive. They have children. And they pass on this new improved version of the TRIM5-alpha gene. Over generations, the old ineffective version of the gene becomes less common, and the new protective version spreads throughout the population. The human genome has adapted. It has learned the face of its enemy. We have taken one step forward in the race. But the race is not over. The virus is still out there, and it is evolving too. Within the viral population, most are still being destroyed by our new defense. But by chance, a few mutant viruses will arise that have changed their structure again. This new variant can now evade our new bouncer. The cycle begins anew. This new virus spreads, putting pressure back on our genome to innovate once more. This is the Red Queen's race. A relentless, cyclical dance of adaptation and counter-adaptation. Every pandemic our ancestors survived was a selective event. It acted as a filter, favoring those individuals whose immune genes, by sheer luck, gave them an advantage. This is evolution in its rawest, most powerful form. So when we look at our own immune system today, we are looking at a living museum of victories against past diseases. The specific shape of your antiviral proteins, the precise way your cells raise the alarm, the very diversity of the antibodies you can produce, these are not random. They are the winning strategies from a million forgotten wars. They are the evolutionary scars, the genetic calluses, built up over eons of viral pressure. The ghosts of ancient plagues are not just present in the fossilized junk of our darklands. Their influence is etched into the very architecture of our most vital defenses. They forced us to become better runners in a race that never ends. Section 5. From Parasite to Partner. We have seen the genome as a battlefield. We have seen the viral fossils as scars from ancient wars, and our immune system as a weapon forged in the heat of a relentless arms race. This picture is true, but it is incomplete. For what happens after the war is over? What happens to the ruins and the relics left behind on the battlefield? The story of our relationship with these viral ghosts now takes a turn that is gentler, stranger, and in many ways more profound. It is a story of reconciliation, a story of how the genome, in an act of breathtaking resourcefulness, can turn an ancient enemy into an indispensable friend. This process is called co-option or exaptation, and it is one of the most creative and wondrous forces in all of evolution. It is the art of repurposing. Nature is the ultimate minimalist. It doesn't like to invent something new when it can find a use for something old. Evolution is a tinkerer, a scavenger, not a grand designer. It rummages through the attic of the genome, through the accumulated junk and the fossilized remains of ancient invaders, and asks a simple question. Can I use this for something? The answer, astonishingly often, is yes. Let us return to the viral fossils, the tens of thousands of endogenous retroviruses, or ERVs, scattered throughout our genetic darklands. We have seen them as broken, silenced invaders. But they are not just silent junk. They are a treasure trove of powerful genetic tools, just waiting to be repurposed. A virus, after all, is a master of genetic control. Its very survival depends on its ability to manipulate the host cell's machinery. To do this, it comes equipped with its own powerful set of switches, dials, and commands embedded in its code. A virus needs to be able to turn genes on and turn them on hard. The signals it uses to do this are its promoters and enhancers, the very same kind of regulatory switches that our own genome uses. A viral promoter is like a powerful engine, designed for the sole purpose of hijacking a cell's resources. Now, imagine one of these viral fossils, an ERV, lying dormant in our DNA for millions of years. It is silent, its own genes broken by mutation, but its powerful promoter switch might still be intact. It is an engine without a car, just sitting there in the junk. Over the vast expanse of evolutionary time, in the endless random shuffling of DNA, this ERV might, by pure chance, land next to a useful human gene. A gene that, for instance, helps the body respond to stress. Suddenly, this human gene finds itself next to a powerful viral engine. The genome, through a random act, has just plugged one of its own tools into a high-powered, foreign energy source. The result can be a dramatic innovation. Now, when the body is under stress, this gene might be turned on much faster or much more strongly than it ever was before, thanks to the co-opted viral switch next to it. If this new, supercharged response provides a survival advantage, the individuals who have it will thrive. They will pass this new configuration, the human gene wired to the ancient viral switch, into their children. The parasite has become a partner. The ghost has been given a new job. This is not a rare, isolated event. As we scan the human genome, we are finding this story repeated over and over again. We are finding that a huge number of our own, essential human genes are at least partially controlled by the fossilized remains of ancient viruses. These ERVs have been wired into our most fundamental biological processes. They act as enhancers that turn on genes in specific tissues. They act as promoters that drive the first steps of development. They have donated their powerful switches to our own genome, becoming an inseparable part of our complex regulatory network. They are no longer just invaders. They have been domesticated, like wild wolves that, over generations, were tamed to become loyal dogs. These ancient viral sequences have been tamed by our genome and transformed into loyal servants. They have contributed to the beautiful complexity that makes us human. We did not just defeat them in an ancient war. We absorbed them. We took their strength, their tools, their ingenuity, and we made it our own. The story of the viral ghosts is not just one of conflict. It is a story of integration, of a deep and ancient merging of two lineages into one. Section 6. The Secret of the Placenta. We have just seen how our genome, like a resourceful scavenger, has learned to borrow the tools of its ancient viral enemies, repurposing their powerful switches to help run our own cellular society. This alone is a profound revelation. But now we must go deeper. For our genome did not just borrow the occasional tool from the viral graveyard. In one of the most critical moments of our mammalian evolution, it did something far more audacious. It did not just co-opt a switch, it co-opted the ghost itself. It took an entire functional gene from an ancient virus and gave it one of the most sacred jobs in all of biology. To understand this, we must turn our attention to the beginning of every human life. We must go to the place where two worlds meet, the place of nurture and protection, the first home we ever know. We must talk about the placenta. The placenta is a masterpiece of biological diplomacy. It is a temporary organ, built by the embryo, that is a bridge between mother and child. It is both a sanctuary and a marketplace. Through the placenta, the mother gives the gift of oxygen and nutrients. Through it, the child sends back its waste. It is a lifeline, but it is also a barrier. It must protect the developing child from the mother's powerful immune system, which would otherwise see the embryo, with its mix of paternal genes, as a foreign object to be attacked. How does the placenta build this magical diplomatic barrier? The secret lies in a remarkable structure called the syncytio trophoblast. This is the outer layer of the placenta, the very front line of contact with the mother's body. And it is one of the strangest tissues in all of nature. It is not made of individual cells. Instead, it is a single continuous gigantic supercell that contains millions of nuclei, all sharing the same cytoplasm, with no cell walls between them. This structure is formed by a constant, astonishing process of cell fusion. Throughout pregnancy, individual cells from the embryo merge with this growing barrier, dissolving their own boundaries and donating their contents to the collective. This fused, unbroken layer is the key to the placenta's success. It creates a seamless shield that the mother's immune cells cannot penetrate, while still allowing the controlled, intimate exchange of molecules. For decades, this process of fusion was a deep mystery. What gave these specific cells the power to merge so flawlessly? What molecular architect was commanding them to dissolve their own walls and become one? Scientists searched the human genome for the gene responsible for this incredible feat of biological engineering. And when they found it, the story of our relationship with viruses was changed forever. The gene was not an ancient human gene. It was a ghost. The protein that directs this fusion is called syncytin, and syncytin is, without a doubt, a viral protein. Its gene is the fossilized, repurposed remnant of an ancient retroviral infection. Specifically, syncytin is the co-opted version of a viral envelope gene. Let us think like a virus for a moment. The envelope gene builds the protein that studs the outer surface of the virus. This protein is the key the virus uses to unlock and enter a host cell. But some viruses have an extra trick. Their envelope protein, after unlocking one cell, can also cause that infected cell to fuse with its uninfected neighbors. This is a brilliant strategy for the virus. It allows the virus to spread directly from cell to cell, creating a network of fused cells like a series of secret interconnected tunnels, without ever having to venture outside into the dangerous open space where it would be attacked by the immune system. Millions of years ago, a retrovirus with this exact ability infected one of our mammalian ancestors. It stitched its code, including its powerful self-fusing envelope gene, into that ancestor's germline. The virus died, its other genes broken by time, but the fusion gene remained, a potent tool lying dormant in the darklands. And then evolution performed its greatest act of creative genius. It saw this weapon of infiltration and repurposed it for an entirely different purpose: creation. The very same gene that a virus once used to create tunnels for its own stealthy spread was now used to build the protective, fused barrier of the placenta. The sword was beaten into a plowshare. The invader's key became the architect of the sanctuary. This discovery is breathtaking. It means that one of the defining innovations of being a mammal, the ability to nourish our young within the safety of a womb, is powered by a gene we stole from a virus. Our very existence, our most intimate biological bond between mother and child, is mediated by the ghost of an ancient pandemic. The line between self and other, between human and virus, dissolves completely in this one beautiful essential protein. We are not just shaped by our conflict with viruses. In some of the most important ways, we are built from their remains. Awaiting continue equals 6, the virus's own membrane with the cell's membrane, prying open a doorway for the viral contents to slip inside. This fusion protein is a powerful tool, and some viruses use it not just to get in but to get around. Once a cell is infected, it can begin to sprout these viral fusion proteins on its own surface. These proteins can then latch onto neighboring uninfected cells, fusing them to the original cell. In this way, the virus can spread from one cell to another without ever having to expose itself to the dangerous, open territory outside, where the immune system patrols. The virus creates a syncytium, a fused network of cells, as a way to hide and propagate. Here, then, were two identical, highly specialized abilities: the placental cells fusing to form a protective barrier, and the virus fusing cells to spread its infection. The resemblance was too strong to be a coincidence. Geneticists began a hunt. They searched for the specific gene in our DNA that was responsible for commanding the placental cells to fuse. They found it, and they named it syncytin. And when they read its sequence, when they translated its letters, a shock of recognition must have washed over them. The sequence was not human. It did not resemble any other gene in the human family tree. Its sequence was undeniably, unmistakably viral. It was the fossilized remnant of an ancient retrovirus's envelope gene. The truth was staggering. Millions of years ago, a retrovirus infected one of our distant mammalian ancestors, writing its code into their germline. The virus died, its other genes broken by time, but this one gene, the gene for its powerful fusion protein, remained intact. And our genome, in an act of pure evolutionary genius, silenced this gene in every part of the body except the developing placenta. It had tamed the viral beast, harnessing its self-fusing power not for the spread of disease, but for the creation of life. The invader's key, designed to break into the house, was repurposed to build the nursery. The ghost in the machine was tasked with rocking the cradle. Without this ancient act of genetic piracy, the secure, nurturing placenta of humans and other mammals might never have evolved. Every time a human being is born, it is thanks to a secret whispered to us by a virus, in a language that we have only just learned to understand. Awaiting continue equals 6. Section 7. Wiring the modern mind. We have just witnessed an act of biological alchemy. A viral gene, forged for invasion, repurposed to build the sanctuary of the placenta. This is a story of a single spectacular act of co-option. But what if this was not an isolated incident? What if the genome's partnership with these viral ghosts is far more extensive, far more intimate, than we have imagined? We now turn our journey from the origins of the body to the greatest mystery of all, the origins of the mind. The human brain is the most complex object we have yet discovered in the universe. It is a network of nearly 100 billion neurons, connected by trillions of synapses, capable of creating art, science, love, and this very conversation of quiet curiosity. And yet, when we look at our genome, we find no simple answer for its existence. We do not have a vast suite of unique brain genes that other primates lack. The bricks and mortar, the basic proteins that build a neuron, are largely the same. The difference, it seems, is not in the ingredients but in the recipe. The magic lies in the regulation. It is the fantastically intricate and subtle symphony of when, where, and how strongly our shared genes are turned on and off during the delicate dance of brain development and function. If the genome is an orchestra, the human brain is its most complex and demanding symphony. So where did our genome find a conductor capable of orchestrating such a masterpiece? A bold and beautiful hypothesis is now emerging, and it brings us back to the heart of our story. The answer, it is proposed, may lie scattered throughout the vast viral graveyards of our genetic darklands. Let us think like an evolutionary tinkerer again. Imagine you want to upgrade a simple electrical circuit into something far more complex. You don't need new light bulbs, the genes. You need new switches, new dimmers, new timers, new logic boards. You need a vast supply of new regulatory components that you can wire up in novel combinations. Where could the genome get such a massive sudden shipment of new parts? The answer is the ERVs. Our genome, through millions of years of ancient pandemics, has accumulated tens of thousands of these viral fossils. And what is an ERV? It is a self-contained package, already equipped with its own powerful promoters and enhancers, the very switches and dimmers that evolution needs. Furthermore, many of these viral fossils still contain the broken remnants of the machinery that allowed them to copy and paste themselves into new locations in the genome. They were jumping genes. Now, picture the scene in our primate ancestors. A burst of viral activity. A new family of ERVs spreads through the population, stitching thousands of new copies of itself into the germline. Our genome is suddenly flooded with thousands of new powerful genetic switches. At first, this might be chaotic, even harmful. But over time, the genome's own taming mechanisms begin to silence them and harness their power. A viral switch might, by chance, land near a gene involved in neuron migration. Suddenly, that gene has a new set of instructions, causing it to turn on in a different part of the developing brain, or at a different time. Another viral switch lands near a gene that helps neurons form connections, giving it a new subtlety, a new layer of control. Each of these events is small but multiply it by thousands across the entire genome over millions of years. The result is a combinatorial explosion of regulatory possibility. It's like taking a simple village with a few dirt paths and overnight, paving it with a dense, intricate network of superhighways, roundabouts, and interchanges. This new infrastructure allows for patterns of traffic and connection that were impossible before. This is what scientists now suspect happened in the lineage leading to humans. The massive repeated waves of ERV invasions provided the raw genetic material, the regulatory parts, needed to build the brain's uniquely complex wiring diagram. These ancient viral switches were co-opted and woven into the very fabric of our neural development, allowing for the fine-tuned orchestration that gives rise to things like language, abstract thought, and self-awareness. The idea is breathtaking. It suggests that our capacity for poetry might be, in part, a consequence of a retrovirus that infected a distant ancestor. The neural architecture that allows you to contemplate the stars might have been wired using the repurposed control panels of an entity whose only goal was to replicate. The line between invader and innovator blurs into non-existence. The ghosts in the machine are not just rattling their chains in the attic. They may be sitting in the control room, their ancient switches now forming the very console upon which the symphony of human consciousness is played. Section 8. A Pandora's Box of Disease. We have been telling a story of beautiful reconciliation. We have seen the genome as a wise and resourceful master, taming its ancient enemies, taking their weapons and turning them into tools of creation. We have seen how viral ghosts help to build our placenta and wire our minds. It is a story of conflict leading to harmony. But this harmony is a delicate one. It is a truce, not a final victory. The ghosts may have been domesticated, but they have not lost their memory. And the covenant our genome made with these ancient invaders came with the profound and lasting risk. For in keeping these tens of thousands of viral fossils within us, we have opened a Pandora's box. As long as the box remains closed, as long as the ghosts are kept silent and asleep, they can be partners in our biology. But what happens if the lock rusts? What happens if the box creaks open? Our genome spends a tremendous amount of energy keeping these viral lands silent. It has evolved sophisticated chemical locks, molecular guardians that patrol these regions, ensuring they remain tightly wound, dormant, and unread. Think of them as sleeping dragons, chained in the deep dungeons of our DNA. In a healthy young cell, the chains are strong, the guardians are vigilant, and the dragons slumber peacefully. But as we age, as we are exposed to stress, to illness, to environmental toxins, these silencing systems can begin to weaken. The guardians can become weary, the chains can begin to rust. And in the quiet of the cellular dungeon, a dragon may begin to stir. A viral fossil dormant for 1,000,000 years can awaken. And when it does, it can unleash the memory of ancient plagues upon the modern body. One of the most devastating consequences is the birth of a traitor within. Imagine an ERV that awakens in a cell. The cell's machinery, seeing a piece of DNA that has suddenly become active, does what it is designed to do. It reads the code. Even if the viral fossil is broken, it might still contain the instructions for one or two intact viral proteins. The cell begins to produce these ancient foreign proteins. Now, picture your immune system, the ever-vigilant police force of your body. Its patrols come across a cell that is producing a protein they have never seen before. It is clearly not self. It is a foreign signature. The alarm is sounded, and the immune system does its job with ruthless efficiency. It attacks and destroys the cell that is producing the foreign protein. But the problem is that cell is you. It is one of your own cells tricked into producing a viral ghost. The immune system, in its righteous duty, has just declared war on a part of its own body. This is the genesis of autoimmunity. Conditions like multiple sclerosis, where the immune system attacks the protective sheaths of our own neurons, are now being linked to the reawakening of specific ERVs. It is a tragic case of mistaken identity, where the body is forced to fight the ghosts that have risen from within its own DNA. The reawakened ghosts can cause chaos in other ways. Remember the powerful viral switches, the enhancers and promoters that were repurposed to run our own genes? That partnership only works as long as we are in control. If an ERV's own promoter switch reactivates on its own terms, it can act like a rogue engine. If that ERV happens to be sitting next to a human gene that controls cell growth, the gene that should only be whispered, the powerful viral switch can jam it into the on position, shouting grow, grow, grow without end. This is a classic pathway to cancer. The ghost is no longer a partner. It has become a saboteur, hot-wiring the cell's most dangerous machinery. And what of the brain, with its delicate, virally wired symphony? What happens when ghosts awaken there? Introducing static into the orchestra. There is growing evidence that the inappropriate activation of ERVs in the brain may be a contributing factor to complex neurological and psychiatric conditions, like ALS and schizophrenia. The very elements that may have helped to build our complex minds may, when they misfire, contribute to their unraveling. This is the dark side of our inheritance. The viral graveyard is not a peaceful place. It is a Pandora's box of latent threats. We are the custodians of a library that is filled not only with our own stories, but with the bound and chained copies of our most ancient enemies. Our health depends on keeping those chains locked. This new understanding is opening up whole new frontiers of medicine, as we seek to understand not just how to fight invaders from the outside, but how to soothe the restless ghosts that already live within us. Section 9: Reading the Evolutionary Scars. We have journeyed through the light and the shadow of our viral inheritance. We have seen how ghosts can be partners in creation and agents of disease. But you may be wondering, as you lie here in the quiet, how do we know all of this? How can a scientist look at the shimmering, almost infinitely complex text of the human genome and find these faint ancient stories? How do we read the evolutionary scars? The process is a beautiful kind of detective work, a forensic science that spans millions of years. It combines the raw power of modern computing with the deep, patient logic of evolutionary biology. The scientists who do this are not just biologists. They are codebreakers, historians, and trackers, following the faintest of trails back into the deep past. The first step is simply to find the fossils. This is a task of pure pattern recognition. Imagine trying to find every mention of a single, forgotten author's name in a library containing millions of books, most of which are written in a language you don't understand. This is the scale of the challenge. Scientists write powerful computer algorithms that are trained to hunt for the specific signatures of a retrovirus. The algorithm scans the billions of letters of our DNA, looking for those telltale genetic skeletons. It searches for the ghostly remnants of viral genes, like the envelope or the reverse transcriptase. But most importantly, it hunts for those ornate, repetitive bookends, the long terminal repeats or LTRs. When the algorithm finds a stretch of DNA that begins with one LTR and ends with a matching one, it flags the region. A potential fossil has been unearthed. Once a fossil is identified, the next question is, how old is it? A geneticist can date an ERV with astonishing cleverness. They do this by exploiting the very process of the viral heist itself. Remember that when a retrovirus inserts itself, its two LTR bookends are perfectly identical, letter for letter. But as soon as that ERV becomes part of our germline, it becomes subject to our own rate of mutation. Like the rest of our DNA, it will accumulate tiny, random spelling mistakes at a slow, roughly predictable rate over the eons. So, a scientist can take the two LTRs from a single fossil and compare them. They count the number of differences, the number of letters that no longer match. If the two LTRs are still nearly identical, it means the viral invasion happened relatively recently in evolutionary terms, perhaps only a few 100,000 years ago. But if the two LTRs are riddled with differences, it speaks of a much deeper antiquity. By counting the mutations and knowing the average rate at which they occur, scientists can create a molecular clock to estimate that this particular virus entered the DNA of our ancestors, say 30 million years ago. Now the story gets even more interesting. We can compare our collection of viral fossils with those found in the genomes of our primate relatives, chimpanzees, gorillas, orangutans. This is like comparing the family records of different branches of a great family. If we find a specific ERV in the exact same location in our genome, and also in the genome of a chimpanzee, what does that tell us? It tells us that the original viral invasion must have happened in the common ancestor of both humans and chimpanzees, before our two lineages split apart more than 6 million years ago. We have just found a scar from a pandemic that predates humanity itself. But what if we find an ERV that is present in the human genome, but is completely absent from the chimpanzee genome? This is the signature of a truly human pandemic. It means that the viral invasion happened after our lineage diverged from the chimpanzee line. This is a ghost that haunts only us, a scar that belongs to our branch of the family tree alone. Section 10: The Viral Tapestry of Us. Our journey tonight has taken us to the very heart of who we are, to the core of our biological identity. We began with the unsettling image of a ghost in the machine, an uninvited author scribbling on the pages of our personal story. We have followed the trail of this ghost through time, witnessing its ancient battles, its domestication, and its potential for both creation and chaos. And now, as we prepare to let these thoughts drift away into the softness of sleep, we must ask one final question. Are these viral echoes truly other? Or have they, through their long and intimate coexistence, simply become part of what it means to be us? The story we have uncovered dissolves the simple boundaries we like to draw. We think of ourselves as a fortress, a pure sovereign self, defending against a hostile world of others. But the lesson of the viral ghosts is one of radical interconnectedness. It tells us that our fortress walls are permeable. Our very foundations are built, in part, from the remains of the invaders we fought so hard to keep out. We are not a pure single entity. We are a mosaic, a collage, a living, breathing ecosystem. Imagine a beautiful ancient tapestry, woven with threads of 1,000 different colors. The main pattern, the bold and central design, is the story of our human lineage. But look closer. Woven into the very fabric of this tapestry are other threads, of a different texture, a different hue. These are the viral threads. In some places they look like scars, remnants of a tear that has been mended. In other places they have been so artfully integrated that they form a crucial part of the pattern itself, adding a depth and complexity that would be missing without them. You cannot pull out the viral threads without unraveling the entire tapestry. This is the image we should hold of ourselves. The 8% of our DNA that is of viral origin is not a foreign contamination. It is part of our weave. It is a testament to the idea that life is not about purity. It is about resilience, adaptation, and the creative power of integration. Our genome did not just survive these ancient pandemics. It absorbed them. It learned from them. It took their stories and their strengths and wove them into its own. We carry the ghosts of 1 billion years of life within us, and their whispers have shaped our song. To know this is to feel a different kind of connection to the world, a deeper and more humble sense of our place in the web of life. It connects us not just to our human ancestors, but to the entire teeming world of viruses that have co-evolved with us, dancing the Red Queen's dance for eons. It reminds us that we are not above nature, but are a living, breathing archive of its grand, messy, and beautiful history. The self in this new light is not a noun. It is a verb. It is a process of constant negotiation, a conversation between our own genes and the ghosts they have accumulated. You are the current living result of that millions of years-long conversation. And so, as our story comes to its gentle end, Let this final thought be a soft place for your mind to rest. The ghosts in your machine are not there to be feared. They are your history. They are a part of your complexity. They are the threads that connect you to a past so deep it defies imagination. The quiet, humming activity within your cells tonight is a symphony composed by countless authors, a tapestry woven by countless hands. Let the sheer wonder of this idea be a final, gentle lullaby. Let it carry you now into a peaceful, deep, and restorative sleep. There is nothing more to learn, nothing left to analyze. Only the quiet acceptance of the beautiful, complex, and haunted masterpiece that is you. May your dreams be peaceful, and your rest complete. Good night.